Yeah, good morning, children. Okay, today we are going to learn about blood groups. So, we already studied about the animal tissues, in that blood is a fluid connective tissue. We already know the red blood cells are there, white blood cells are there, blood platelets are there, blood plasma is also there. These all we learned. One more interesting thing we are going to learn. So you know that you are thinking that your siblings and your family members, you think that your blood relatives, but it is wondering to know. So in the world, many people are connected with us, with our blood. Many people are there, the same blood group. Actually, real blood relatives are there on the globe everywhere. So here we are going to identify the blood groups with the help of kit. Okay, children. Here, if you observe carefully, there are 4 blood groups are observed by the scientist name Carl Landsteiner. Carl Landsteiner in the year 1900. 4 blood groups: A, B, AB and O groups. So in that he observed that these all the blood groups. How he observed? He collected many blood samples of the different people and he mixed the different blood groups, blood samples, and he observed in some blood samples, he observed that there is a clotting of blood, but in other samples there is no clotting. So by this he observed that, so there are 4 different groups are there. There will be A positive, B positive and AB positive and O positive. By having RH factor, he also observed that if RH factor is present in that, he said that the positive group. If RH factor is absent, that blood groups are known as negative groups. Understand, children? There are 4 groups he observed: A, B, AB and O. In that, some of the groups are positive and some of the groups are negative. Why they are negative and why they are positive? He observed one factor in the blood, that is RH factor. If the people with RH factor, they are known as the positive groups. The people without RH factors, they are called as negative groups. Total world, in the, for example, you take 100% of people, in that 93% of people with RH factor and only 7% of people without RH factor, without RH factor and with RH factor. 93% of people with RH factor. So by this he did this all the divide the blood groups in this way. Now we are going to do this activity by knowing of our blood groups. Okay, children. So how many of you having this blood group A? Just raise your hand. Do you know your blood group? How many knows your blood group? Those who are A blood group, once raise your hand. Yeah, you are all my blood relatives. Why? Because my blood group also A positive. Understand, children? Okay, now we are going to do this activity. How to identify our blood group? First you observe the table. From this table we can understand which group is positive and which group is negative by knowing. Okay, so we have anti-A, anti-B and anti-D. These are the 3 substances we have. Yeah, start. Make 3 circles in each. In anti-A, we can identify these blood groups by, so for example, we adding anti-A in the first circle, and if it is get clot, and there is no clotting here, no clot. Here again clotting we observed. This blood group is known as A positive. And next we take anti-A is no clotting, clotting of B and clotting of RH factor. So it is known as B positive. Next we can see clotting in anti-A, clotting in anti-B, and clotting in all three. So AB positive. Next we see there is no clotting in anti-A, no clotting anti-B, only clotting in anti-D. That group is called O positive. Now you see that we can see in the anti-A clotting, no clotting in anti-B, and no clotting in anti-D. This blood group is A negative. Next we can see no clotting in anti-A, clotting in anti-B, and anti-D also no clotting. Then this blood group is known as B negative. Now we can see clotting in anti-A and anti-B, no clotting in seen in the anti-D. That blood group is known as AB negative. And final, there is no clotting anywhere. This is O negative. So these are the blood groups observed by Landsteiner. In this way we can easily identify the blood groups. Now we are going to do that activity. Okay, in this AB blood groups are called as universal recipients, and the O group are called as universal donor. We can identify this blood group in this way by seeing table. Very easily we can understand. So here you observe that this is clotting, no clotting, doesn't clot. So by this we can easily identify the blood groups. Now we are going to do that activity. What is the aim for this activity? So this aim for this activity to identify the blood groups by the help of blood identification kit. So now procedure. What is the procedure we have to follow? First we require one porcelain plate, and in that we have to make 3 circles. Mark as a anti-A first circle, next anti-B second circle, anti-D third circle. Now we require blood identification kit. It containing of 3 serums, anti-A, next anti-B serum, next anti-D or RH factor we required. Then we need surgical spirit and needles we required, and a cotton. The needles are disposable. We use only once for every person, and cotton balls with surgical spirit. So what we have to do first? We select any one person and squeeze the blood and prick the finger and come anyone. Identify the blood group of D.
Akshaya.
Akshaya. Okay, Akshaya, be ready. So first you take surgical spirit. First you will clean the finger. Any one finger, clean the finger. Hold it. Now you take disposable syringe. Disposable needle. So you just prick the finger. Now you collect the one drop on first circle and second drop on the second circle and a third drop on third circle. Close. Now you add a drop of anti-A for the first drop. Next you have to add a drop of anti-B serum to the second drop of the blood, and third you have to take anti-D or RH factor serum to the third blood drop. Now you take a needle and mix it thoroughly, mix it properly, and then observe, and you clean the needle very clearly. It should not mix to the other drop of the blood. So now you mix the second circle and next wipe it properly, and then you go for the third drop of blood in the third circle, and then just wait, just mix it properly, thoroughly, then you can identify the blood group. See, have you seen any clots here? In anywhere? No clots.
No clots.
Your blood group is what?
Oh, positive.
Wo?
Positive.
Positive.
Yeah, because
so
there are
he-
no clots in my blood.
See here. What is here? Is all clots. Here? What is this? Positive, positive. Where you send the positive? Is O positive. Can you see the clots here? Yeah, exactly. Yes. So slowly now we are observing clots in the RH factor, anti-D. We can clearly see in this. See in the A, there is no clot. In the B there is no clot. Only in the RH factor, anti-D, we can see the clot very clearly here. So by this we can identify that the given sample is
O positive.
O positive. So here